Good morning, class. I welcome you to Metric Hub. Today we are going to talk about DNA profiling. DNA profiling, let me just quickly go into the subject so that we can answer other questions that are below in order for us to understand the subject or to understand the topic in detail. Let's begin. I'm going to define what is DNA profiling. DNA profiling, or it can also be called DNA fingerprinting. It is a technique used to analyze a person's DNA fragments. It's a technique that is used to analyze a person, to analyze a person's DNA fragment, to analyze a person's DNA fragments. That is DNA profiling, a technique that is used to analyze a person's DNA fragments. So in this regard, what we are saying is that we have heard of scenarios and life experiences where people or criminals, perpetrators of various cases has been found as a result of DNA profiling or DNA, which we are saying the DNA is used as a way of identifying certain individuals who might have done certain criminal offenses. So it is through DNA samples. So what we are saying is, what is it? What are the sources, or where is this DNA found from? So I'm gonna just show you to say sources of the DNA samples, or where can that DNA be found from? Sources of the DNA samples from a human body or from individuals or from what? What we are saying is, where is the DNA found? We want to understand to say the samples of DNA can be used as ways of leading in terms of investigations, in terms of solving some unresolved disputes that might take time or that are so complicated. So in this regard, we are saying, what are the sources of DNA? What are the sources of the DNA that is used in terms of the systems that we are referring to? So the sources of DNA, we can talk about the saliva. The DNA sample can be found from the saliva. It can be found from the hair. It can be found, the DNA can as well be found from, we are saying saliva here. It can also be found from the semen. It can as well be found from skin tissues. Which can be skin cells. Just to mention, but a few among other lists. So what we are saying is that the DNA is collected from the hair. It is collected from as evidence from the saliva. It can be evidence from the semen, as for in cases of maybe saying rape cases, we can talk about the skin tissues, which can be found hanging on some items that might be found in some places. And then just to make this one again, to make it clear, we also have DNA sample as from the blood. I think this one we mustn't just leave it out because it forms a key part also in that respect. So samples or DNA can be found from what? From the saliva, from the hair, from the semen, as well as blood samples. So how does this process take place? How do we do DNA profiling, or how does DNA profiling goes? How is it done? I want you to understand that we have got certain nucleotide segments of the DNA do not code for proteins and exist as repeated short sequences of bases. So in that respect, what happens is that DNA of a person is obtained through such samples that I've identified there, and it is cut into fragments using a specific enzyme that is used. A specific enzyme is used to cut the DNA fragments of a person that has been identified, or I mean, the sample that has been found. These DNA fragments are separated and arranged to form specific what? To form a specific pattern or bands. The specific bands or patterns are then arranged. I mean, the DNA are arranged, so therefore they form a specific what? They form specific bands or patterns. In that case, we are having what? The different patterns that you can see there. As you can see, we are saying the DNA fragments that have been cut into, I mean, the DNA has been cut into fragments using specific enzymes. And then what happens is that these enzymes are separated and arranged to form what? Specific bands, which are as shown in this diagram that we are saying there. Diagram below, which was used or which was produced, DNA sample, and then we have got different people who are given there, Bato, David, Jane, as well as Tabby. We are saying these are different samples of DNA that we found. And one thing that you should understand is that each person has got his own DNA or has got his own set of DNA, except in cases of identical twins. Identical twins can have specific matching DNA. So let us quickly just answer these questions, and then we can move forward in this case. So I've given you to say those are the sources. Let's quickly answer the questions. The first question is saying, I mean, the scenario given there is that detectives were investigating a crime scene and found blood on a broken window. They suspected that the blood was that of the criminal. To identify the criminal, they analyzed a DNA sample from the blood and compared it to that of 4 suspects. The diagram below was produced. So a DNA sample of blood was found on a broken window, which we say that that was the crime scene. So when cases or when there are scenarios where police need to solve they have to solve criminal cases, they usually collect or they have to use DNA samples in cases where the perpetrators are not actually being found directly. So the DNA samples are collected. In this regard, we are saying the blood sample is the one that was found that was on a broken window, which would be also be used as the what? As for the suspects. So you've got the DNA sample that was the DNA sample. We are saying this is the sample, which is written DNA sample. It is actually representing the DNA fragments or the one that was found on the crime scene, the DNA sample. And the 4 people who are given there, we are saying these 4 individuals, we have got 1, we have got 2, we have got 3, and then 4. All of these people, we are saying their DNA is being picked or is being used as what? As suspect. It has been analyzed and then it has to be compared to that which was found on the DNA, I mean on the crime scene. So let us quickly answer these questions. The first question is saying, name the technique that was used to identify the criminal. Question 2.1. 2.1.1. Name the technique that was used to identify the criminal. What we are saying is, what technique was this one? We are saying it has to be known that this is what? DNA profiling. This is DNA profiling, which we define that the DNA profile or the DNA is a technique that is used to analyze a person's DNA fragments. So the DNA fragments which were found there, they were then shown there as you can see. So we are saying the technique that was used there, it is what? DNA profiling. DNA profiling, question 2.1.1. Question 2.1.2. Question 2.1.2. The questions reads again, who is a possible criminal? Who is a possible criminal? So in this regard, we are saying we have got the DNA sample that is being given here, and then we have got the variety of individuals, 4 people that are possible suspects here. In this case, who is a possible criminal? We are saying when the sample is compared to this, the DNA sample on the crime scene is compared to these ones that are given there, who is a possible criminal? The one that looks alike, or that looks that resembles, or that is more likely to form or to match the sample that was found on the crime scene, is the one that is for the possible criminal. So in this case, we are saying if you look the one for battle, we see we have got this, it can't match here. We have got 2 bars and we have got 1 bar here. There's different David, and then we are saying we have got Jane. But if you can see the one for Jane, we are saying the first, we have got a single bar there, double fragments there. We have got 1 there, we have got 2 there, we have got 2 there, and then the 1. So if you can check the DNA sample for Jane's DNA sample, it matches the one of the crime scene, which was found on the crime scene. So it is what? It is Jane who is the possible criminal. It is Jane. Jane is our possible criminal in this scenario. He's our possible criminal. Explain your answer above. Why do we say that Jane is a possible criminal? That is question 3.1.2, question 2.3.1. Why do we say that it is? Explain your answer. How can we say it is Jane who is a possible criminal? Why should we say that? We are saying Jane's DNA profile or Ben's matches the DNA profile from the crime scene. The DNA profile of Jane, as you can see there, we say that it is a match. Jane's DNA profile or Ben's matches that which was found on the crime scene, that which was found on the crime scene, which was found on the crime scene, which was found on the crime. It is similar to that which was found on the crime scene, the DNA profile or bands of Jane. Hence, we are saying he is the possible criminal here, found on that, similar to that which was found on the crime scene. As we can see, we are saying that if you can check here, we have got the double bars here, we have got the double bars there, we have got a single bar here, it is also having a single bar there, we have got 2 bars here, which is also similar to this one. We have got 2 bars there. We also have 2 bars there. And then eventually we are saying there is 1 bar here, which is also a 1 bar there. So this is actually showing that this is showing that this DNA sample or this DNA bands or profile that was found for Jane is a possible criminal as a result of the matching bars, the matching profiles that are found there. Our next question is saying question 2.1.4. Question 2.1.4. 2.1.4 is saying question state 1 other use of the DNA. State 1 other use of the DNA of the technique identified in question above. So the technique that we are saying is what? DNA profiling. What is the other use? What is the uses? So we just need to list the uses of this one. So the first use is what? Is to identify possible criminals. That is one, to solve criminal cases or to solve criminal matters. That is one of the things to say we can use it in that regard. So the other uses, we are saying DNA profiling can also be used as for proof of paternity. One, it can be used as proof, it can be used as proof of paternity, proof of paternity. Paternity, we are talking about the possible father of a child, people, they father children, men, they father children, and then at certain times they refuse responsibility or they refuse the fathering of those children. So we can use it as we can use DNA profiling for solving paternity disputes, proof of paternity. What else can we do, or what else can DNA profiling be used for? We can also use it for tracing missing persons, tracing missing missing persons, tracing missing persons. That is very correct to say that sometimes people, they go missing. I guess we have heard uh, either on the different media platforms, which can be either from the radio, it can be from the TV, it can be from newspapers. Utatawako, we have talked about Kumbulaikaya, such types of people who are going missing or who cannot be traced or who cannot be found. We can be able to confirm and attest that these are the real people missing when their DNA profiles or when their DNA has been collected and has been compared to other family members or siblings of the same family of those individuals. We can also use DNA profiling in identification of genetic disorders. It can be used for identification for genetic disorders. Uh, Identification of genetic disorders. I'm sure that there are certain diseases that are family related that are passed from generation to generation. As in, like in saying, we have got individuals who suffer from hereditary conditions, which to say that these conditions, they have inherited them from their parents. So in terms of identifying such problems or such diseases, such kind of disorders, uh, the medical field can as well use DNA profiling for identifying such congenital or those are inheritable diseases or conditions, that is genetic disorders that can be identified there. We can also identify dead bodies or dead persons and animals. Identification of dead persons, dead persons or animals. Dead persons and or animals. They can as well be identified through DNA profiling. They can be identified through DNA profiling. We can as well to say, we can also confirm the genotypes of plants and animals. Confirm the genotypes of plants and animals. As to another point that we can give there. Confirm genotypes of plants and animals. I'm sure, class, you remember and you understand what we mean by what we mean by genotype, we said that is the genetic makeup, the characteristics that are within the blood that defines an organism, which are giving the structure or the physical appearance, the functioning, and as well as the appearance of an organism is defined by the what? By the genotype. So what we are saying is that plants and animals genotype can also be confirmed through genetic engineering, through DNA profiling. Last but not least, we can also use it for matching tissues for organ transplant. This is number 6. I just want to give a full listing. Identification of matching tissues for organ for organ transplant, organs transplant. I can see you understand that we do organ transplant or organ transplant is done also in hospitals, in the medical field. So in order to identify the ones which are matching, we are saying again that there is need for DNA profiling that can be used in that respect. Let's just answer the last question, the last part of our question. I also just used that to say the DNA profile that we have seen here, it is a different scenario whereby you can see that this is an investigation case of a criminal case. I mean, a criminal case was being resolved in this regard or in this respect. But at the same time, we also said again, the uses of DNA profiling, which is question 2.14, we have given those ones, which number 1, which we have said solving paternity disputes, which is this one on under the uses paternity disputes, which have already prompted me to bring a question which is related or which is related to that situation of paternity disputes or paternity problems. We have got question 3 here, given that Tom and Maria have 3 children. One of the 3 children was adopted. A DNA profile for each member of the family was prepared to determine if Tom is the father of all the 3 children, which is the 3 children, Annie, Mary, and Steve. The DNA profiles are given below. These are the DNA profiles that are given below there of the 3 children of Tom and Maria. So the question reads, which one of the children has been adopted? Which one of the children has been adopted? So in this case now, we are saying the DNA profile, it is supposed to match the DNA profile of the child should be showing a resemblance of that of the what, similar to that of the parents. So what we are saying is that which one of the children has been adopted in this case, which one has been adopted, which must show that the DNA profile should show a resemblance of the parents there. If the person or if the children or if the child is born from the mother and father who are the same there as Tom and Maria. So which of the children has been adopted? In this case, we have got Annie, we have got Mary, we have got Steve. If you look at Annie's DNA profile, we can see that we have got this bar here for Mary. We have got this bar here, which is also similar to this bar here for Annie. We have got this bar here, which is also similar to this bar here, which is similar to the father. And then we have got this bar here, or bent here. It is also benting similar to that of the mother. If we look at this one of Mary, Mary, we are saying if you look at the band for Mary, it is totally something opposite. If you can look at it, you see the DNA bands for Mary. If you see these bands, this bar here, we cannot say that it can be, we cannot say that it can match to this one. It cannot match to that one. We go to this one, we come to this one here. Either we can say it might be like saying it want to relate to this one, but not exactly. But we have got a tiny bend here, which must be on the top for the mother here, but it cannot match there. And then finally, we have got this bend, which is at the far bottom, which is also not showing there. However, we can look at Steve. Steve, we are saying the one for Steve, Steve is having a smaller bar here, which can also be a replica of the one for Mary. We can mark it there. We have got this bar here, this one here, which we can also give that it is still representing Mary there. And then we have got this bar here, which can also be a replica or an identification of saying it is showing that for Tom. We have got the last small bar here, which we can as well give to say that it can be representing that one of what? Of Tom. So the adopted child here, we can give to say Mary, number 3.11, 3.1.1, Mary is or shows to be an adopted child. Mary shows to be adopted. Which one of the has been adopted? Mary can be the adopted child. And then 3.1.2, 3.1.2, Mary can be an adopted child. And then explain your answer there. As you have said there, we are saying it doesn't match to either of the parents. It cannot match the DNA. We are saying because the in this case of Mary, we are saying if you can look on that one, because they all matching bars, patterns are not showing there, we can say that she's a possible adopted child because none of her DNA is not matching to that either of the father or what of the mother. So reason is because no matching bands or bars to either of the parents. No matching bands or robust to either of the parents. That's the reason why we can say that she can be the adopted child. So as you can see, we have given this, and I'm sure you've seen how do we calculate that. I've shown you the calculation that I've been showing, that I've been trying to mark with different pens there to say that the pens for Mary are not matching to either of the parents, hence she can be the adopted child. No matching pens, bars, patterns, or DNA profile with the both parents. Both parents does not match to a DNA profile or to a pens or to a bars. Hence, she can be a suspect or she can be identified as the adopted child. I'm sure you've seen this topic. I've came up with these different questions, so therefore we can be able to relate and show the different angles that the question might come in the exam. I thank you so much for your time. Do not forget to follow or do not forget to subscribe our channel, Metric Hub, and you always see that our information will remain posted, or keep on being posted and keep on being updated as we go. All the best. Tell your friends, you tell another friend that life science has been simplified through Metric Hub. Let's meet and let's change our life. We can have level 7 in Metric. That is quite possible through the revision interfaces, in revision programs, revision exercises, revision session that we are going to run on Metric Hub. All the best, and please keep subscribing and following the channel for more information. And thank you very much. Good day.